and he and his wife are running this organization, and in a very professional and a wonderful way. And सौरभ जी, हमारे अरविंद वर्चस्वी जी जो है हमारे चेयरमैन हैं ऑफ आयुष कमेटी, इनकी फर्म का नाम है श्री श्री तत्व. ही इज वेरी क्लोज टू गुरुदेव श्री श्री रविशंकर जी.
Pleasure to meet you here, Dr. Arora ji. It's a pleasure. Thank you.
Same here. Very nice.
Okay, जी, whenever you are ready, we can start. पहले बाकी सारे पैनलिस्ट आ गए हैं.
Good evening, पंकज जी, कैसे हैं?
वो जरा कंफर्म करो तो then let's start.
Pankaj ji is also joined, sir, from NABL.
Very good afternoon to all of you, sir.
नमस्कार पंकज जी.
नमस्कार सर. नमस्कार सर. मुल्तानी सर.
नमस्कार.
I think
Dr. Arora, very good evening.
Very good evening, sir.
Very great to see you here after a long time, actually.
Yes, sir. Same here, sir. Very long time. I think because of Corona, we're all not been able to interact as much.
Yes.
And I think, if I'm correct, Dr. Shastri ji has made it mandatory for all AYUSH laboratories to be NABL accredited.
Not me, the absolute tab.
But has that been implemented, Dr. Saab?
The notification will be out in a day or two.
Okay. But the one thing is that Dr. Shastri ji, while he's Pankaj ji, of course, you know Dr. Shastri very well. Apart from being a very, very knowledgeable person, he takes the perfect decisions in life. And I think whatever he is trying to do, I think even Saurabh ji will agree. I think that's a very, very appropriate and the correct decision. All the decisions which the Ministry of AYUSH is doing are absolutely outstanding.
In the morning also, we had this discussion in the senior officers.
Ji.
And we got the appreciation because my question is very simple. The Ayurvedic doctors are going there, and the analytical chemist who is standing there, if he puts a question, which kind of HPLC column is required for C3 and C4? Could they be answered? Not even one in the country, and it's not possible also, I don't expect. So they have no right to certify these labs. That's my point.
Very true, Dr. Shastri. Absolutely, perfection.
FSSA on the other side has allowed NABL accreditation as a mandatory certification to recognize a lab. Then why can't we? That's it.
Wonderful, wonderful, Dr. Shastri ji. I think all of us, including Dr. Saurabh Arora, Dr. Apna Hamare Sodhi ji, Arvind ji, we all welcome that. Absolutely outstanding.
Appreciating what, sir?
Yeah. Vivek ji, 4 baj gaye bhai.
We are just upgrading Mr. Tripathi. He's joined as an attendee, sir. Just give us half a minute.
Okay, perfection.
Host, please please upgrade Mr. Arvind Tripathi. He's in the attendees list. Karamjit, please do that.
Dr. Shastri, आज त्रिगुणा जी की बड़ी भाभी जी का ना, she unfortunately expired today, so she will not be joining us.
Okay.
So he gave me a call in the morning around 11ish, so he won't be joining. और Dr. Katyar के भी बेटा is in a hospital.
I know that.
हाँ जी, so he will also not be joining.
I guess that's taking time. So, with your permission, can we start?
Please, Dr. Shastri ji ने हरी झंडी दी, तो please go ahead.
All right. So, a very good evening to all of you. A fateful evening yet again, I would say, not only for our webinar on testing protocols and stability studies on AYUSH products. It's also fateful because we are currently running, while we speak, probably the first ever vaccination camp for the members of any chamber and their families and the employees of our members and their families. So there are about, I think, more than 300 vaccinations which have happened since morning. So this is our small effort to make our environment and our ecosystem go back to normal. So therefore, I am so pleased to also have this webinar today which is again a very interesting topic, considering testing is going to become only more important by the day, considering the fact that we are going to the international markets with our AYUSH products. So without much ado, we have, of course, our chief guest for today, Dr. JLN Sastry, the CEO of NMPB. He needs no introduction. We have with us our Deputy Director from NABL and the industry speakers, along with our Senior Vice President, Mr. Pradeep Multani, Shri Arvind Varchaswi ji, our Chair of the AYUSH Committee at PhD, and Mr. Jitinder Sodhi, Co-Chair of our AYUSH Committee. So I'll request our Senior Vice President, Mr. Pradeep Multani, to kindly give a welcome and presidential remarks. Over to you, sir.
Vivek ji, the sharing option needs to be activated.
For the presentation, Dr. Sastry?
Yeah.
Yes, we'll give you the right, sir.
Good.
Admin, can you please do the needful?
Good evening to everyone. On behalf of PhD Chamber, I would like to welcome Dr. JLN Sastry, CEO, National Medicine Plants Board, Ministry of AYUSH, Government of India, and Dr. Pankaj Goyal, Deputy Director, National Accreditation Board for Testing and Calibration Laboratories, NABL. I would also like to welcome Dr. Saurabh Arora, Executive Director, Analytical Division, Arbro Pharmaceuticals Private Limited, Dr. Arvind Tripathi, Manager, Quality Control, Hamdard Laboratories, India, and Mr. J.P. Sinha, Vice President, Analytical Division, Multani Pharmaceuticals Limited. We also have with us Mr. Arvind Varchaswi, Chair, AYUSH Committee, PhD, CCI, and Managing Director of Shri Tattva, Mr. Jitinder Sodhi, Co-Chair, AYUSH Committee of PhD, CCI, and MD of AYUSH Herbs Private Limited, Mr. Arjun Multani, Co-Chair, AYUSH Committee of PhD, and today's session moderator, Mr. Vivek Sehgal, Assistant Secretary General of PhD Chambers. Ladies and gentlemen, PhD CCI has been working as a catalyst for the promotion of Indian industry, trade, and entrepreneurship for the past 116 years. It is a forward-looking, proactive, and dynamic pan-India apex organization. As a partner in progress with industry and government, PhD Chamber, with a special focus on MSMEs, works at the grassroots level with strong national and international linkages for propelling growth, harmony, and integrated development of the Indian economy. Over the centuries, many medical systems have emerged from Asia. Today, there is an increasing interest in Ayurveda, Siddha, and Unani systems of medicine for prevention, diagnosis, and treatment of an extensive range of ailments. Interest in these medicines is particularly by those who have benefited from the holistic approach from previous treatment and by those who have apprehensions concerning the toxicity and safety of drugs. To have the efficient efficacy and safety, it is required that the formulation should be stable. Stability means ability to remain unchanged, but it is common experience that all objects, including formulations, changes or deteriorates with time and undergo reactions after specified periods. To enhance the quality standards and maintaining stability, the focus on testing protocols and stability studies assume much greater significance. The present volume of protocol for testing of Ayurveda, Siddha, and Unani product contains various parameters of testing for different categories of single and compound formulations, limits for heavy metals, microbial load, pesticide residue, aflatoxins, and methods for determination of these safety and quality parameters. It is known that potency of ASU preparations reduces after a certain period of time. To have a higher efficacy and safety, it is mandatory that the product should be stable physically, chemically, microbiologically, and therapeutically, as well as free from toxicity. A well-designed stability protocol containing information such as selection of batches and samples, test attributes, analytical procedures, acceptance criteria, storage conditions and period, testing frequency, sampling plan, and various types of stability study and stability testing methods should be taken into consideration. Currently, pharmaceutical product is generally assessed using a validated stability indicating analytical method and an expiry date mark based on the predicted period from date of manufacture, when the pharmaceutical product would show more than 10% deterioration in the active molecule. Stability guidelines have also been implemented on Ayurvedic formulations where percentage degradation can be assessed when the product is stored at different conditions of temperature and humidity. The general concept of stability for Ayurvedic or modern medicine remains same, but the parameters used to assess the stability may vary from product to product. The benefits of stability testing include concern for well-being of the patient. The manufacturer, by ensuring the product quality, helps in selection of adequate and appropriate formulations, excipients, and container closure systems for a product to determine shelf life and storage conditions. It also ensures preparation and substantiation of the claimed shelf life for the registration dossier and to confirm that no variation occurred in the formulation or the manufacturing process that can adversely affect the stability of the product. Today, we have dignitaries from Ministry of AYUSH, Government of India, and industry to deliberate on the importance of testing protocols and stability studies of AYUSH products, and we urge the industry to learn more about it and comply on the same. We also have experts from NABL, and each of these departments is more like a role model and a teacher to us. And we encourage everyone to contact them, learn from them, and get more informed on the subject. On behalf of PhD Chamber, I once again welcome you all and seek your active participation in this discussion. Thank you. Jai Hind and God bless everyone.
Thank you. Thank you very much, Senior Vice President. It's great that you have set the tone for this important webinar. And we will make sure that during this very important session, we get some very critical information on testing protocols and certification from Dr. Sastry and the other experts. May I now call upon our chair of the AYUSH committee, Shri Arvind Varchasvi ji, to kindly give his remarks. And we have been told by Dr. Sastry that he will be here with us till 4:55 p.m. because he has a meeting with the secretary at 5 p.m. So right after this this talk by our chair, we will be inviting Dr. Sastry for the presentation. So over to our chair, Shri Arvind Varchasvi ji.
Thank you so much, Vivek ji. Dr. Jailen Shastri, CEO, National Medicinal Plant Board. Dr. Pankaj Goel, Deputy Director, National Accreditation Board for Testing and also Calibration Laboratories, the NABL. Of course, my very good friend, Mr. Pradeep Multani ji, our Senior Vice President. Mr. Sonal Repsanyal, Mr. Jitendra Sodhi, our co-chair, and a very warm welcome to the other panelists for the session, Dr. Saurabh Arora, Dr. Arvind Tripathi, and also Mr. J.P. Sinha. Traditional medicine is widely used in prevention, diagnosis, and also treatment of an extensive range of ailments. Over the centuries, many medical systems have emerged from Asia, and today there's an increasing interest in Ayurveda, Siddha, and Unani systems of medicine. Ayurveda is a sacred system of healthcare which originated here in India, whereas Unani medicine originated in Greece and was introduced to India by the Arabs. Stability testing is necessary to ensure the product is of acceptable quality throughout its entire storage period. In order to do this, it is necessary to monitor the compliance of the product with a suitable quality specification throughout the shelf life. Quality consistencies of drug products are essential to guarantee expected therapeutic activities, and achieving consistent qualities of herbal products is challenging because of their physiochemical complexities and inherent variabilities. Regulatory authorities worldwide have issued regulations or guidelines for stability testing parameters and testing procedures for herbal products stored in proposed conditions. These testing parameters and methods for finished herbal products are detailed in guidelines and regulations issued by 5 global authorities in 15 countries. The stability of finished herbal products can be determined by testing for properties susceptible to storage conditions, which include physical properties like organoleptic characteristics, physical condition, particle size, the chemical properties like the assays and the active compounds, the pH levels, the identification, etc. And also with regards to microbial and also toxicological properties. These properties can all affect the quality, safety, and the efficacy of the herbal product, and thus the shelf lives of the herbal products should be determined by stability testing. Furthermore, different stability protocols are used in different countries, as herbal products are generally developed to meet national guidelines. Global harmonization of stability testing has been recently emphasized upon in the context of herbal drug development. But the adoption of these international standards can only be achieved by sharing national experiences and information. The WHO Expert Committee publishes technical reports annually on specifications for pharmaceutical preparations and guidelines on good herbal processing practices for herbal medicines and on stability testing for active pharmaceutical ingredient and finished pharmaceutical products. The stability depends on various factors like nature and the product itself, the ingredients, the packaging material, and the environment condition. Stability testing is a complex set of procedures as a variety of factors influence the stability of the product. The formulations itself are complex in some cases, and the degradation reactions may be complicated by possible interactions of several components of the formulation. So, ladies and gentlemen, having said that, on behalf of the PhD Chamber, I once again welcome all of you and look forward to your active and valuable participation in this important session. We have with us Dr. JLN Shastri, a repository of great wisdom on the science, and especially when it comes to stability, and also on the aspect as to how an Ayurveda product or service really needs to be treated. I look forward to listening to him on the testing protocols and the stability studies as the subject that what we have here today. Thank you very much, Mr. Vivek.
Thank you. Thank you so much, Chair, for those encouraging remarks, as well as giving out some details and setting the outline for the webinar on testing protocols and stability studies. Time for us to hear our chief guest, Dr. JLN Shastri. And as our chair rightly put it, I think he's an ocean of knowledge, and time for us to dip into it. Over to you, Dr. Shastri.
Dr. Shastri, you might have to unmute yourself.
Yeah, I'm sorry. So thank you very much, Vivek, Mr. Vivek, and respected Chair Arvindji, then Vice Principal of the PhD Chamber for AYUSH Committee, Mr. Pradeep Multani, and respect other members. Yeah, it's a privilege to be a part of this discussion today. And indeed, it is an important aspect, as we all know. Without spending much time, I will put the things in right perspective. And looking at two aspects. So one is the testing protocols. The second one is the stability.
Dr. Sab, you are muted. Dr. Sab, you are muted.
No, I'm trying to move my slides. There's a problem.
Okay. And
trying to move my slides.
And Dr. Shastri, we are happy to tell you 174 participants are attending this program today.
Oh, great. So just let me. Are you able to see my slides moving or not? That's the only question.
Not as yet. Not as yet.
आपकी वो पहली स्लाइड ही है टेस्टिंग प्रोटोकॉल्स एंड स्टेबिलिटी स्टडीज ऑफ आयुष प्रोडक्ट्स डॉक्टर JLN शास्त्री.
Give me 1 second. I will unshare and again share it. Are you able to see it now? No.
No, Dr. Shastri, not as yet.
Vivek,
Admin, can you help? And
One second. I'm able to get it connected again. You will
Okay.
get it now. Hope you are able to see this now.
yes, we can. And you can do a full screen. Yes, absolutely fine, sir. Now we can see.
Right. So as we all know, we as industry are governed by the Drugs and Cosmetics Act 1940, which has 5 chapters and 2 schedules. And then when it comes to number, and 1945 have 19 parts, and except that you have all the alphabetals for which you have schedules. And importantly, among the sections, we look at only mostly the section 33, which speaks about adulterated drugs, spurious drugs, and even misbranded, where the standards, one way or other, are embedded because under section 33 EB, if you do not have your drugs as per the standards to be penalized, and section 33 EC, even as a central authority, I can also issue a government order saying that this particular product can be banned without the involvement of the state licensing authority. And one interesting thing which we notice most of the times, which needs to be corrected, and we are in the process, is section 33H, where the provisions of your drug inspector, everything is extended to section 23, 22, 24, and 25 of chapter 4. Whereas section 33A clearly speaks that this section, the chapter 4 is not applicable to ASU drugs. So therefore, it needs correction. It gives ambiguity, and therefore needs correction, and the upcoming amendments will address this. Coming to the rules, again, there are very minimal rules. 151 to 170, altogether 21 rules, 20 rules, in fact, are going to speak about it. And mostly the rules which are concerned with the raw material part and the standards rules, and then the application for license and the conditions thereof, and rule 158. These are things which are important, and we are looking for the standards where from they have emerged. So section 33, then 153, 160, and then your 161 for compliance on the label, and so on and so forth. So in this, the main topic which we are discussing today comes from rule 164, where the method of test or analysis which is to be employed for these materials, whether it's raw material or the finished product, it will be decided. And the standards have been laid as per rule 168. That's how the system goes, and Schedule T, which speaks about the GMP norms, which speaks about your detailed testing laboratory, and so on and so forth. And each rule is having a link with the form which you are applying. In Form 25D, if you apply, then you get your license in E. And once you have applied for a loan license, it goes in 24E. Now we are adding E1 as another form, which will give the ease of business, because we are also trying to identify the 3P license. Along with loan license, there should be 3P license that is being addressed. Then certificate for your good manufacturing practices comes from 155B. Now, the drug standards. So what are the standards that basically we are talking about? These are basically taken care by the pharmacopoeia laboratory, then your CCRS, they are also helping pharmacopoeia laboratory. More than 75,000 applied formulations and 750 ASU drugs, which the ASU drugs have the pharmacopoeia standards and the pharmacopoeia part 2. And then proprietary, they should have the in-house standards. That's how it's decided. But there are certain problems as we look at these formulations. In the text, you don't have the clarity on weight by volume, volume by volume, or weight by weight. across this, and this is where the industry should help. Recently, we had a meeting, and tomorrow morning at 10:30 we have the final meeting. We'll come back to you. It is industry which has to drive this, because to make a batch size, these are very important, and legally also they're very important. You are preparing Singanarugu glue using gum and then trifluoroquada and then a powder. So and you have sulfur also. So when you have used different items, one is by volume, one is by weight, it should be clearly mentioned in the formulation. Otherwise, many places these problems will come up. So that needs to be addressed, and industry should share the mechanism of batch size and the wastage in each batch. That's how we need to come together and work on this. Then the equipment used. We may have to give some kind of precision, though we may not be specifying the companies. At least we should specify the instruments, and this needs revision. Then the AYUSH premium certification and standards by QCA is another thing which has helped in the standards, and therefore the government supplies many times as questioned industry. I'm taking a lead for you. The L1 or Q1 concept that needs to be addressed, and we have a mechanism in place there. And you know the ASHRI tab for the Ayurveda Siddha Unani the Technical Advisory Board, and this will be fortified with homeopathy team also, because homeopathy is now going to be part of your chapter 4A. The amendments are on. The Pharmacopoeia committee is actually on the PCMNH is going to decide. Mostly, I'm talking here about Ayurvedic, but there are Unani, Siddha, Homeo, Pharmacopoeia also. So these are the Pharmacopoeia. I don't want to waste the time. Part 1 has 9 volumes, and part 2 has about 5 volumes right now. And you have formulary 3 volumes, and 4th volume is ready for the veterinary medicine. And about 645 different monographs are there. And Atlas has been also released. These are the basic standards which have been defined by Pharmacopoeia, and formulary speaks about the formulations from the classical text, and this is the PLIM which is working on this. We have roughly about 45 testing laboratories certified. And what are the different categories of the material which we will be usually handling or testing? The raw material, the intermediates, and finished goods. So when I speak about raw material, this is known. Again, the formulations doesn't have a raw material always. So this is something which we have not focused ourselves. Like for example, in Chandraprabhavati, one of the formulations has your camphor. Camphor is not a raw material; it is an intermediate or a value-added product. If we are using a chandan tail in some formulation, it is a value-added product. You can't call it as raw material. So using khair in some of the farmers, katha, which comes from again the forest department and laboratory from Rajasthan. Nobody else can manufacture also. So it is an intermediate. It's not a raw material. And Gataria oil, volume 6, which starts with the volatile oil, is not a raw material. So these corrections are in the process right now. Otherwise, in the international scenario, we will face difficulties. So accordingly, the pharmacopoeia needs to be reorganized, and is happening. That's how the Rule 158 is amended, and the notification will be out maybe tomorrow, maximum by Monday. So where the extracts have been considered as intermediates. Or take the example of Chyawanprash. Many of the manufacturers who are manufacturing Chyawanprash, please look at it. It's not a single formulation. It's a combination of 3 intermediates which have been prepared in a syrup base to make it a linctus. What does it mean? You are preparing pishti, you are preparing decoction, you are using prakshapadravya. You have 3 intermediates which are finally being processed in your sugar syrup. There is no jaggery in any formulation of Chyawanprash in Ayurveda. So when you process the syrup-based formulation, and it's a linctus. So this is how we have to categorize and reorganize our formulations, and the process we have started recently. The finished goods, as you know, classical formulations as per part 2 of API, and proprietary patent in-house standards. Now, these in-house standards are very important for industry, because all your exports are basically depending on these particular category of products. So the question is, what are the quality standards and the protocols for raw material? Okay, because the pharmacopoeia starts with the raw material. So you have several pharmacopoeial monographs where I would like to give this. Mostly across the globe, these are the major aspects. Though there are a lot of other things, I want to take the top 10, which are the main things. I'm not not going to talk to you about antibiotics, which is being discussed with the US Pharmacopoeia, AYUSH, and Pharmacopoeia Laboratory for a long time. That requires time. Even I was part of IORA discussion 2 years back with CMAP Lucknow, where we could not draw any conclusions. But there also I have given the concept of fit for purpose. All the 42 different kinds of antibiotics, we need not just like that for fancy test it. Let us come to a common point. Even tomorrow I have a similar discussion with BRICS countries on the AYUSH part. So there also we want to, we have done some kind of harmonization looking at all these regulations, some common minimum program and fit for purpose. Since I have a Mercedes Benz, I won't take it to bring bricks and cement bags, and none of us will do it if we are wise enough. So let us use the same common sense when we look at any standard, whether it is a herbal, mineral, herbo-mineral, or as a matter of fact, modern chemical drugs. The science remains the same. There's no change in the science. So here, the description, which is a major part, and then they should all go by only the botanical name. That's I just submitted my concept on one standard to Secretary AYUSH, and maybe we will see limelight very soon. When the source used comes from same soil, same geoclimatic zone, why Ayurveda, Siddha, Unani, Homeopathy, USP, IP, BSP, all will have different standards. If one have one standard, you do grades. You can make it as grades. Today, paracetamol USP grade, paracetamol IP grade, paracetamol BP grade is available. Make it such a simple thing going forward. And then moisture, ash value, water soluble extract. Moisture has a lot of big role to play. I will show in my slides. And then minimum TLC oblique HPLC, GC, ULC, whatever you want to do. Today, people are crazy to get to that kind of thing. So microscopic description will always give a lot of important information which we cannot ignore. Like if you take the roots of Piper longum and Piper retrofractum, the medullary rays are 5 and 6 respectively, and such a simple difference. Okay, and you, by no chemical means, usually you will be in a position to differentiate. However, you say the piperine and all, and being root, you will not get larger quantities of piperine, negligible quantities of piperine. So you should have a fit for purpose. Again, with this simple sectioning, if you are able to test it, why do you want to go for costly techniques? That's exactly what we need to do. Okay, and then the microbial load, heavy metals, and aflatoxins. These are protocols. Just I don't want to again bore you. These are the standard protocols which are available with AYUSH, and there are the website. And many times you might have seen the flow charts. Yeah, it's a systematic approach, having your standardization profile, then you have your SOP for extract, then your protocol for the formulation into which the extracts are getting into, the kind of combinations you are making. Again, here one important aspect, we always take it granted that if you have 4 or 5 herbs, just take all of them and extract them and use it. Please don't do it. Ayurveda never said this. Not even modern science. In the childhood, we read about, maybe in the 7th or 8th standard, we read about the difference between a mixture and a compound. Let us keep it in mind, and it gives a lot of important information. While doing some work on AYUSH 82, we noticed it. While working on AYUSH 64 recently, we have noticed taking 4 ingredients and making extract, the chemistry is entirely different from using them individually as a mixture. So don't take things what you are going to assess when you have a standardization protocol and when you have the safety efficacy evaluation along with that. The biological screening can be in vivo, in vitro, that's up to you. And the pharmacopoeial entry comes for those products which are safe. We all are aware of it. And then this is how we have organized the raw materials in the part 1. Right, from botanical description, the physical description, the identification, whether it's macroscopic or microscopic, and TLC or HPTL, maybe HPLC, GC in case of volatile oils. And those are the things, and physicochemical parameters. Loss on drying is also very important. Sometimes you use a stage at which you received it. This is always going to be a crucial factor. And in a formulation like Chyawanprash, if your Pipli has 1% moisture variation and you compromise, your company, if they look at their profit and loss account at the end of the financial year, so it will penalize you. And as a matter of fact, you take one of your major products on that 1%, if you have about 10 crores turnover, 1% to 1.5% moisture. So you are selling water in place of the product. The efficacy will come down, and the your so-called wastage is you are comparing water with some kind of kesher or chandan. So that's the kind of mistake you will be doing, and it has a lot of repercussions and heavy metals and all. Now, this is the protocol again, which is available with us, and the flow charts, they're available. Okay, so these are available on the website. I will just go through these slides, therefore you are aware of it. So the SOP for the finished product, where all the standard ingredients are mixed as per the formulation, and then it will be tested as per the guidelines. And again, in this also you will be looking at mostly the what you call whatever the major component ingredient you have. For that, you will be having markers. Like, for example, again, I take Dashmul Arist or Ashok Arist or Arjun Arist and Aswa Aristas, and Avalehas, I will take Ashwagandha Avaleha, and then your Chyawanprash again. We have to select a few of the ingredients and trace them. Otherwise, it's very tough in polyherbal combination. That's why US pharmacopoeia, the botanical drugs, of course, recently I heard the definition has been changed, but otherwise they don't allow more than 2 herbs because highly possible. We could convince US FDA when they visited when I was in industry, and they agreed on the argument, and they did not ask for the details of all the ingredients of Chyawanprash. So, but polyherbal formulations, it's up to you. Otherwise, your team, if they miss major ingredient like kesher, how will you come to know about it? If you don't have a kesher standard in your so-called Chyawanprash, of course, in some of the formulations is not there. So you have to identify saffronol. What is the percentage of saffronol? What is the percentage of sandalol? Because sandalwood is also added to that, and this your so-called hydrolyzable tannins percentage because you have 24.62% of your Chyawanprash as amla. So you can do that. But while testing, again, I'll tell you, the Chyawanprash testing method given in the books may not work for you. You take any Chyawanprash, you get the same TLC. The problem is, we have not realized that we are hydrolyzing it because either hexane or petrolate you take, and then you process it because fish tea is having ghee and oil, and it's gone. Rather, I prefer to take the solvent which was used in the petroleum method, and then test it for HPTLC and GC to find out what are the components that are coming, and major components should be made as markers for identifying Chyawanprash. Otherwise, all your Chyawanprash samples will pass. Okay, so we need to. So again, fit for purpose. These tests should stand for your requirement, not fancy these things. And like the pesticide residuals, at least these 9 pesticide residuals and testing, if it happens, is something which is always going to be helpful, whether you have a GCMSMS or LCMSMS. The microbial counts, you're all familiar with that. And the stability part, which is again an important aspect for all of us, and which was introduced in 2016 after a lot of deliberations. You all know the GSR 789, which was given in 2016, has given us 3 years time for the implementation. But at the time, somehow they missed out the accelerated stability. And then with the great difficulty in 2018, we had a meeting, and in 2019, finally, an advisory was issued by the Ministry of AYUSH that accelerated studies will also be part of the called stability studies. These stability studies, they cannot escape from the accelerated as well as the real-time stability studies, because volume 8 of API already mentioned about both the protocols accelerated at 40 degrees plus or minus 2 degrees at 75% relative humidity, and your real-time or the so-called long-term stability, it is about 30 degrees plus or minus 2 and 60 RH. And now these are all on the basis of ICH GCP guidelines. On the ICH GCP guidelines, the stability, whether it is a long-term or accelerated, is undertaken on primary and or your commitment batches. Usually 3 batches is the protocol for which you will generate the data. And if you have any variations in the other formulation or some kind of excipients or anything, again, at least 2 batches resubmission is all that's required, and that's usually all of us are following. Supporting stability data is also important. Specifications of each of these products and the shelf life and shelf life specifications, stability indicating methods. Okay, and then comes the accelerated and long-term stability. See, I will explain further. Okay, let me go to this. So when we look at the stability, WHO actually in its technical document, as was mentioned by Arvind ji, time to time it produces the technical reports. 953 in 2009 it has given. It all depends on the 4 climatic zones of the world where you are going to trade or sell your market your product. That is very important, whether you are doing in a temperate, subtropical, tropical, very high humid, dry areas. So depending upon all these things, you have your you have to have your adjustment to the stability protocols. More or less, as we always talk about, I was mentioning this your real-time protocol also in my previous slides. WHO again made an amendment, and 30 degrees centigrade, and when it is about 65% RH. Now the conditions have been amended about 75% RH, because they say it is always safe. Sometimes the humidity, like this is a general case when you look at APIs. This time I'm taking the API, sorry, ICH document as such, and I'm mentioning it. It may not be relevant to all of our products, but it is essential for all of us to understand these are the standard ICH guidelines. There is no mandatory what you call protocol here. There are protocols, but there's not mandate. These are guidelines which will guide you to the right direction. Whether you put it 27 or 25 is up to you, but within the range. And as was mentioned by Mr. Pradeep Multani, that the 10% variation below that is all that what we are going to look at. And then the second protocol what we have for the active pharmaceutical ingredients, when you are storing it in the refrigerators. This is a little alien concept to us, because we don't handle those products like your injectables and vaccines and other things. But this is also there. When you are using a refrigerator, then also you have protocols. Just I'm running through. So the commitments, what will be made most of the times, as I mentioned, are 3 production batches on the basis of which you will generate the data and submit to the licensing authorities or the regulatory authorities. Okay, and this is applicable to finished products also. This is exactly what I'm trying to tell you. The accelerated stability in many cases is of prime importance for the industry, because even with 1 month, even GCP guidelines clearly mention about it, even with a 1 month report, you can file an application. That is the kind of advantage what you have. And because it takes time when it comes to modern drugs, though we many times say that it takes more time for us with drugs, but the time taken for the modern drugs is much more than what we see. So then, if you have 3 months accelerated stability, at least you can have 1 year shelf life. You can launch your product, and subsequently, once you complete 6 months, on that basis, you can claim 2 years real time, that shelf life, sorry. And then by the time 2 years complete, you may get your real time completed also. So that's how you can manage your stability studies. If these long-term studies are conducted, as I already mentioned, the other minor variations of and that's what the finished products are all about. Then when it comes to the package, which is another important aspect along with your finished product, you have to always think about it. Take an HDP container, take a glass bottle, take a plastic container or a poly container, and then look at what kind of things, like you have a new generation of products, new formats for which you should have a proper thinking, like your new packs where you have multi-layered packing system. And then if you want to put your juices there, how are you going to handle it? So it's all up to the team of your scientists who will finally give a kind of a protocol, though there are plenty, as I was mentioning earlier, for these finished product also your refrigerator and freezer protocols. Okay, so these guidelines for stability testing in the pharmaceutical products which are containing these substances in a conventional dosage forms. So what are those things? So the mostly to select the adequate formulations and container closure systems. The type of study most of the times is accelerated study, and this is used in the development of the product. When to determine the shelf life and storage conditions, accelerated as well as real time. To substantiate the claim of the shelf life, the real time is very much required. To verify that no changes have been introduced in the formulation, then accelerated in real time, where quality assurance in general, including the quality control, will be associated with the process. So I was mentioning about these 4 zones. In fact, this slide should come in the first instance, like temperate, subtropical, hot and dry, and hot and humid. These are the 4 zones. Depending upon these zones, you have to decide what are all the protocols you are going to use. This is the amendment WHO has given. And to design the stability, these are all ICH stability study and design protocols. You can get it, test samples, and your studies accelerated and this thing. Then frequency of testing, then analytical methods used, and this is the stability test summary sheet, which is there in ICH guidelines, and your stability report. And then finally the storage. Okay. I want to simply conclude saying that the normal storage conditions which have been defined by WHO is storage in dry, well-ventilated premises at temperatures 15 to 25 degrees, or depending on climatic conditions up to 30 degrees. That depends on the relative humidity. Or can be considered as normal storage conditions, but it varies from country to country. Okay, and we have a lot of publications. Just this is one always which attracts the mind, Mohammed Sajid Ka. And I will show a few examples. These are from my previous industry experiences. We have done some study when I was in Dabo on some of the raw materials. And the main change what we found on most of the raw materials is the moisture loss and drying. Otherwise, as per the technical document, you will write it as LOD. And this is going to be a major problem. So therefore, we should know what are the raw materials which requires humidity control in its storage, because that was the main problem. Okay, and in the list, not all herbs have problem. Like bubble is a little variation, but not the major variation. Shankapushpi, the variation is very high, we have noticed, and Mustak, the variation is very less. We do not have any variation. So all these things need to be assessed, and we need to address this by storing these materials under humidity control. It may be cost, but if you want to make your product more stable, we want to give it longevity and as per standards, it may be required. This is another product for which is a simple suspension, antacid suspension, where mostly the pH was the concern, nothing more, nothing less. And then another product here, if you see, okay, the tannins and alkaloids have been taken and they have been regularly checked at various intervals. These are some of the examples I'm showing. When I was concerned this paras, which is sold to rackets and bankers, we have done some studies, and we need not be very stringent. You should pick up some parameters because you can't define everything. So it's up to you. What kind of standard, in-house standard you are going to generate, that's up to you. So apart from this, just I want to emphasize on the clinical protocol, because the safety efficacy in the end is going to decide whether the standards are going to really have something or not. Therefore, somewhere during the development of the product, you should also get focused on the good clinical practices. The guidelines have been issued by Ministry of AYUSH in 2013, 31st March. So I would like to conclude by saying that testing protocols and stability protocols are very simple. There's a 10-point protocol if you keep with you, that is sufficient. The description, moisture, ash value, which is again total as well as acid insoluble, to look at whether any contamination from heavy metals is there or not. Then water soluble extractive, alcohol soluble extractive value, your TLC, HPLC, HPLC. Then microscopical descriptions, then microbial loads, then heavy metals and aflatoxins wherever it's necessary. Then your stability protocol, as per already the discussions happened, which is there in PI, the real time and accelerated, whichever is there, and you can follow that. So with this, I conclude my presentation. I thank PhD Chamber of Commerce for giving this opportunity and looking forward for interaction. Thank you.
Thank you so much, Dr. Sastry. I think a beautiful presentation, and I think the way you concluded it, with a 10-point program, it reminds me of, I think, one of our former prime ministers of the country who had actually had a 10-point program to monitor the progress of the country. So very well put, Dr. Sastry, and I'm sure almost 200 people who are listening to you would have grasped a lot of information and knowledge from this particular presentation, sir. It's so kind of you to share with us all these details, especially with the manufacturers who are actually looking at conquering, I would say, the international markets. In the same flow, now time for me to invite our speaker from NABL, our Dr. Pankaj Goyal, Deputy Director from NABL. And I think his perspective will only add on to what Dr. Sastry said, especially from an international perspective, because NABL is again a part and parcel of QCI, which is a World Council for Standards. So it'll be great to have the talk from Dr. Goyal. Over to you, sir.
Thank you very much, sir, for giving this opportunity. And sir, before proceeding, could you provide me the sharing option for this presentation?
Sure, please. So can the admin please give him the sharing rights immediately? Thank you, Paramjeet. Thank you.
I hope my slides are visible to you now.
Not the slides, but it's just saying, yes, now it's visible. Perfect.
Okay, fine, sir. So very good afternoon to all of you present in this webinar. Honorable Dr. Shastri sir, CEO National Medicinal Plant Board, respected Chair Mr. Varchasvi ji, respected Mr. Pradeep Multani ji, Senior Vice President of PhD Chamber, and all the eminent personalities of the day present in this webinar, Mr. Sinha from Multani, Mr. Saurabh Arora from Arbro, Dr. Tripathi, Mr. Sodhi, and Mr. Vivek Saigal. Firstly, on behalf of NABL, I welcome you all and for this webinar on testing protocols and stability studies on AYUSH products being organized by PhD Chambers of Commerce. Here I am Dr. Pankaj Goyal, Deputy Director NABL. Going to present on NABL accreditation and its importance with particular aspects of the AYUSH products. As in previous, presentations, Shastri sir has already detailed a lot about the, AYUSH products, its related, pharmacopoeia, that is API, and all the 10 points protocols which are actually requiring for the stability studies. That includes both chemical and microbiological studies. And of course, once we are going through this 10 points protocol, the AYUSH products which we are going to produce, they would have the quality product in terms of the international market. My slide is not moving actually. So I will start this from this quotation that quality is never an accident. It is always the result of an intelligent effort, said by Mr. John Ruskin. So definitely so far we are talking about the quality and we are talking about the stability of the products of the AYUSH, and so that we can give a common person the quality products, whether it is in the Indian market or in the international market. But for getting the quality products, we should have the intelligent efforts, and definitely our regulatory boards, including the National Medicinal Plant Boards and other regulatory boards of the AYUSH Ministry particularly, they are doing very intelligent practices in providing the quality products in terms of the AYUSH. For this, if I'll go for the general guidelines of the drug development for the AYUSH formulation by the CCRS, Ministry of the Commerce. So the section 11 is clearly says that quality control section will have a minimum of, I'll directly go to the part 2, that manufacturing unit shall have a quality control section, which has been explained under section 35(2). Or alternatively, these quality control provisions will be met by the getting testing, etc. from the recognized laboratory for Ayurveda, Siddha and Unani drugs, as per the under Rule 160A of the Drug and Cosmetic Act 1940, which sir has already explained in the previous presentation. So if I'll go for this rule number 160A, that is the application for the grant of the approval for testing Ayurvedic, Siddha and Unani drugs, it clearly says that about the application for the grant or renewal of the approval for carrying out the test for, these are the 4 parts, that is the identity, purity, quality and the strength of the Ayurvedic, Siddha and Unani drugs. So ultimately, when we are going to test whether it is the raw material or the finished product for their identity, purity, quality and the strength with the 10-point protocols which have already been described by Shastri sir. So we are going to get a good stable product in terms of its authenticity and the purity, as well as about the disintegration, that if anything is going to happen for the AYUSH product in terms of the long-term study, after the long-term studies. Not even the in Indian use, but also for the directions are available for the export point of view, in which it has clearly that Government of India, Ministry of Health and Family Welfare, Department of Ayurveda, Yoga and Naturopathy, Unani and Siddha Homeopathy says that one directive has been issued to the manufacturer of the Ayurvedic, Siddha and Unani drugs to test the drugs manufactured and exported by them for the presence of the heavy metals and make declaration accordingly on the labels. Along with this order is there. So these are the practices or the intelligent efforts are being carried out by the Government of India and the Ministry of Health and Family Welfare, so that we can get the quality product, not for the Indian point of view, but for the international point of view. And ultimately, here comes the picture of NABL, that but with a quote again, that without a standard, there is no logical basis for making a decision or taking the action. So we are doing the 10-point protocol or other things, but if we are not under the good standard which can denotes our competency, then this all things which we are doing in terms of the stability protocols or the other studies, that would be all in vain. So we should have one logical basis. We should have a good standard which will denotes whatever we are doing, that is absolutely right. So enable comes in the picture with this, that authenticity of the test results, whatever we are doing, this increases if these testing laboratories are also accredited by NABL in accordance with ISO IC 17025, which is emphasizing significantly on the test witness, competency of the personnel, metrological traceability and calibration of equipment, and ensuring the validity of the results by proficiency testing and the quality management system. So a glimpse of ISO IC 17025 2017, that is has been given, in which there are the other requirements from scope to other. I'll basically focus on this clause number 5, that is the structural requirement, clause number 6 resource requirement, and clause number 7 process requirement, which actually gives an insight that when a person will enter into the laboratory, what kind of the structure should be there of the laboratory in order to pursue any kind of the testing activities, what kind of the resource requirement should be there, including the personnel, the facilities and the environmental condition, equipment and their metrological traceability, as well as process, under the process requirement, whatever the method you are going to use for the validation of the other studies, there should be proper selection and the verification and the validation. In a blank procedure, it should be completely validated and verified properly, so that whatever you are going to produce the results, it will be acceptable throughout the world. It is further emphasized by the evaluation of the measurement uncertainty and ensuring the validity of the results as well in terms of the ILC, interlaboratory comparison, and the proficiency testing. So according to ISO IC 17000, that is conformity assessment vocabulary and general principles, it says that accreditation is nothing but the independent evaluation, that is third party attestation related to any laboratory that has been termed as conformity assessment body, CAB, which is conveying its formal demonstration of its competency to carry out specific conformity task. Specific conformity task is nothing but the parameter or the test which you're going to perform as per your required guidelines given by the regulatory board or other pharmacopoeia requirement. So accreditation will give you the formal recognition of the competency, as well as it will ensure the sound quality assurance system, and it will ultimately lead to the enhancement of the customer confidence and satisfaction. So once the customer will be more satisfied with your with the services you are provided or what kind of the product you are being provided, it will also increase the business and market advantages. It will also reduce the technical barrier to the trade and will because of the global acceptance of the reports, and ultimately the laboratory will get the international recognition and definitely increase the business. Indian economy will also be increased, and it will also lead to increase in the staff motivation as well. And definitely the issues, the test reports which are being issued by the accredited laboratories are acceptable amongst all the economies which are signatories to the ILAC MRA, and that means that tested once and accepted everywhere. So here I would like to tell you that NABL is signatory to ILAC and APEC MRA since 2000, and we are continuously being signatory for this. So definitely now come to the National Accreditation Board for Testing and Calibration Laboratory. In short, that is NABL. It is a constituent board of any Quality Council of India, and it has been established with the objective to provide the government and industry with the scheme for the third-party assessment, for the technical competency of the testing laboratories, calibration, medical laboratories, proficiency testing providers, and reference material producers. And we also operates as per the international standard of ISO IC 17011, that is conformity assessment and general requirements for the accreditation bodies, accrediting conformity assessment bodies. And we offer the accreditation services to the laboratory in a non-discriminatory manner, and with the ILAC and APEC MRA as well. This is the international scenario of the accreditation, where the whole globe is under the under the Ilac operation, and in Asia Pacific region this is APEC body, and in India Annual India is working for the accrediting as per ISO IC 17025 and other standard. This is a Indian structure. That under the QCI there would be the NABL, then as per ISO IC 17011, and we are giving the accreditation for these 5 different domains, that is the testing, calibration, medical testing laboratories, proficiency testing provider, and RMP. We have all the domains, like in the previous slides also the sir has explained that 10 points protocols for the chemical and biological testings. So in this testing chemical and biological, we are covering all the kind of the aspects of these 10 points, as well as the toxicological and safety studies as well. This is the accreditation process. I'll not go in detail. It is available in NABL website and NABL 100. These are the general documents which are also available in NABL website. We have accredited so far more than 6,000 laboratories, including testing 3,340. Particularly, if I'm coming for the AYUSH laboratories, it is 48 laboratories in chemical discipline and biological 43. However, in entire drug and pharma products, we have accredited more than 126 laboratories with in chemical discipline and biological 84 laboratories. And we are working to bring all the drug and AYUSH product testing laboratories under the accreditation as per ISO IC 17025, so that ultimately we can provide the quality products. We have the recent development in NABL, that digitalization with providing the NABL portal. That is a one-stop window for the entire accreditation process, online application, and app-based and geotech-based assessments and paperless assessment. We have also initiated the integrated assessment with the regulatory boards to avoid the multiple assessments. Some of the regulatory boards have already joined the NABL, that EIC, FSSCI, APIDA, T-Board, Spice Board, IOP, PC, etc. And so that the laboratory will get advantage of getting the only one assessment and having the certification of all these regulatory boards. So here we can have an option for the AYUSH industry as well, and other drug and pharma regulatory board, that we can have the single assessment for the laboratories in terms of the integrated assessment. So laboratory will get not only the NABL accreditation, along with they will also get the regulatory board of that AYUSH recognition as well. We have also introduced the online e-learning portal platform, that is the Quest. It is available on NABL website. Anybody can enroll there and can go for the e-learning mode. However, there is a lot still to be done by way of articulating the challenges that have until now discouraged the pharma and AYUSH laboratories from addressing the benefit of NABL ISO IC 17025 laboratory accreditation. And in this particular regard, we should increase the awareness and the capacity building with respect to the benefits of this international standard against the startup cost and other challenges, given that the concept of testing once and accepted everywhere. And finally, it's a joint call for NABL and relevant regulatory board and laboratory associations as well to ensure an improved level of awareness of accreditation in the drug and pharma sectors, including AYUSH products. So ultimately, we can use the accreditation in order to demand the quality products, and we should recognize the accreditation. We should promote the accreditation. So ultimately, we get the quality products and stable products in terms of the AYUSH and other drug and pharma related products. And for this, thank you very much for your kind attention, and thank you very much to all PhD members for giving me this opportunity. And finally, thanks to CEO NABL, Mr. N. Venkateshwaran, for nominating me for covering this webinar on testing protocols and stability studies of AYUSH product. Thanks a lot.
Thank you. Thank you very much, Dr. Goyal. Wonderful presentation. And I think, again, a lot to grasp for all those who are involved in the quality of products, quality of AYUSH products. I just had one query, a quick one. You talked about certain number of AYUSH labs being accredited on biologicals as well as some on chemical. Is there a number which you may have on in which both these biological and chemical accreditation is being given by any chance? Any overlapping numbers there?
No, sir. Actually, there are certain laboratories which are being accredited in chemical and biological both. So this is those numbers, and some laboratories are only getting accreditation for chemical particularly, or the biological particularly, as per their requirement. That's why the numbers are being different in the chemical and biological.
But there are certain laboratories which are both accredited for biological as well as chemical.
Yes.
Any numbers on them you may have by any chance?
I'll let you know for that, sir.
Okay. Anyway, thanks a lot. Thanks a ton for sharing that information. And
Thank you very much, sir.
thank you, Dr. Goyal. Time for me to invite our co-chair of the AYUSH committee, Mr. Jitendra Sodhi, for some quick industry remarks. Over to you, Mr. Sodhi.
Very good evening. I would like to welcome Dr. Jailan Shastri, CEO, National Medicine Plant Board, Dr. Pankaj Goyal, Deputy Director, NABL, Shri Pradeep Mutani ji, Senior Vice President, PhD Chamber, Mr. Arvind Vasachvi, our Chair, AYUSH Committee, PhD Chamber, and all the secretaries and all. Ayurvedic medicines have proven to be very effective in many health ailments. Even in the modern era, the popularity of the herbal medicine is increasing day by day. Traditional medicines are system of medicines such as Ayurveda, Siddha, Unani, and homeopathy, based on herbal medicines, have long been cornerstones of health care in India. And now worldwide is awakening, worldly it is awakening the power of these systems. The growing demand of global acceptance of these systems has dramatically changed and the dynamics of industry and the needs of the herbal testing. Medicine and formulations which are earlier made by the folklore medicine traditional Vaidyas to treat patients within their villages and neighborhood now have traveled through the length and breadth in our country and worldwide. These changes and the global reach have put tremendous pressure for quality control of these products, and herbal testing has rapidly progressed to keep up with this pressure. Today, there is increasing demand of herbal testing. Most of the herbal medicines contain multiple active agents. Due to the complex nature of herbal medicines formulations, it has become very difficult to apply standards quality control parameters. The stability depends upon various factors like the nature of the product itself, the ingredients, the packaging material, and the environment condition of that place in the country. It is complex set of a procedure as a variety of factors influence the stability of the product. The formulation itself are complex and degradation reaction may be complicated by the possible interaction of several components of the formulations. The purpose of stability testing is to provide evidence on how the quality of the drug substance or drug products varies with time under the influence of varieties of environmental factors such as temperature, humidity, light, and storage conditions. The protocol for stability testing is prerequisite for starting stability testing and is necessarily a written document that describes the key components of regulatory and well-controlled stability studies. It is important that we strengthen the system of the following the testing protocols and conducting the stability studies. The previous speakers has very well defined all the protocols for testing and stability factors. Like I really appreciate that our chair Arvind ji and Jailan Shastri ji has put a very wide knowledge on this. But there are some suggestions which I like to put in through this. I think that we need to make our herbal suppliers, our mandis, to understand what is the need of the industry. Like they should understand, because the raw material, if it is of a standard, then the outcome of the products will be very standardized. If we get material free from aflatoxins, free from heavy metals, free from pesticides, free from all the microbial loads, then it is though we process it before making the formulations, but then it is a more likely that without those we can make a very good quality product as per the international standards. So I suggest that we should establish such laboratories at mandis and or doorstep of the collectors or cultivators or people who are working in this industry to provide the raw material for the herbal industry. I agree also with Dr. Jayant Shastri. There's a lot of things need to be done in pharmacopoeia. A lot of revisions need to be done, and that is true to meet international standards. I think those are very much needed. We face a lot of problems in international level for our standards. Like there is, I was just giving an example. We have set up, heavy metal limit, lead 10 ppm, arsenic 10 ppm. Wherever in the US, it is not more than 2 ppm. So in arsenic, even it is 1 ppm. So there are in certain states, like in California, it is 0.5. So I think we need to really work on it and then to set up the better standard for our industry to sustain in international market. I'd like to thank you, PhD, and the chance given to me to come up with my suggestions and the industry suggestions. Thank you, Vivekji. Thank you, Multani Saab, that I've been given the chance. And it was a great listening to everyone here. Very knowledgeable. Thanks.
Thank you, Mr. Soni, for giving us a perspective of the industry on this vital issue. Time for me to actually call upon our next young, vibrant speaker, Dr. Saurabh Arora. He's the Executive Director of the Analytical Division at Arbro Pharmaceuticals Limited. So as we were discussing, I think you run a fantastic laboratory, as our Senior Vice President was talking about. It'll be great to hear from you, Dr. Saurabh. Over to you.
Thank you so much, Vivekji. And it's a real honor to be here sharing this platform with stalwarts from the industry and the regulatory people like Jayant Shastri ji, Pradeep Multani ji, and Arvind ji, and Pankaj ji from NABL, Tripathi ji, Sinha ji, Sodi ji. So it's really an honor to be sharing the platform with all of you. And I will be trying to go more in depth towards the practical, actual conduct of the stability and try to do it real fast, because a lot of the points that I'm going to be talking about have already been covered by the experts. I was just trying to make a note of what there is that I will be able to add more value to. So I'll be focusing on this. So first and foremost is, what I'm covering in the presentation is the need for the stability studies. What are the regulatory guidelines that are driving this? What are the prerequisites for successfully doing a stability study? And important things like sample management and data evaluation. How do you really decide whether the study is passing, failing? What's the shelf life you can assign? So why do we need to do a stability study? So all of us know, if I want to get a license, I need to get it done. If I'm registering somewhere, I need my marketing authorization, I need to do it. WHO GMP, registrations for exports. So these could be all the different reasons. But I appeal to the industry that we should all think of it. There's even more important reason. Our company's reputation is on stake. Number of complaints, it can spoil our image, especially with the current social media scenario where even one pack of fungus, growing in a product can really spoil the product brand image totally, and saving you against potential legal action. So overall, giving you peace of mind. So unless we think of it as a value add beyond just the regulatory requirements, I feel we don't want to invest in it as much. It is very important to invest in the stability studies because it will really add value to the products. So this is an example. What do you want your product to be? If you want it to be like a solid block of gold, which will be like a gold block forever, or do you want it to be, rusting pipes, which will cause a lot of trouble for everybody? So I think everybody wants our products to be like gold, value going up and stable as a rock. So when should we do stability studies? When we are doing new products, variants, there are any major changes. And also if there is there's usually a requirement that you should keep on doing it as an ongoing program to monitor your products. What are the regulatory guidelines? Lots of guidelines, ICH, WHO. But for us, I feel the API Part 1, Volume 3, page number 296, I think that's where the story starts and ends. It's a wonderful guideline, really detailed, and it covers everything that we need to do for our stability studies for the AYUSH products. So first thing which comes to mind when you talk about a stability study is I need a stability chamber. So there are a couple of types of chambers that you can go ahead and buy. So you could go for a reach-in chamber. These are usually smaller, cheaper, or you can have walk-in chambers. They have their own advantages. They reach in Almira kind of chambers. They are cheaper initially, but if you go with a walk-in one, they are cheaper in the long run because the per unit storage, running cost, maintenance, and all is much lower. So I personally recommend you go with a walk-in type. And these chambers should have temperature, ideally plus minus 0.5 because the ICH recommends that your chamber should be within 0.2 and humidity should be plus minus 2% because the ICH says you should be able to control it at 5%. So when you will qualify and validate these systems, do the calibration, temperature mapping, it will pass. How many do you need? So for India, you need 2 as per API. You don't need to think too much. Get a 4075 and a 3060. And that will do your requirement for accelerated and long run. But if you're in the global markets, exporting to other countries and have different packs, permeable containers or hygroscopic samples or low temperature storage products, maybe like soft gelatin capsules, you would need more conditions and you'll need to decide on that. So a quick buying guide for the stability chambers, go for all SS, go with temperature data monitoring, which is computerized. Have a system which has got standby cooling, heating, humidity, SMS alerts, alarms, and now you can even get split systems where the refrigeration can actually be put outside and the chamber can be kept inside. Go with a trusted vendor and discuss the qualification protocols because when your audits will happen, client audits will happen, they'll be looking at the qualification documents. For trouble-free operation, you should have a AC room, split operation, good quality water, and regular preventive maintenance of your stability chambers. Next, now you have your chamber in place. You want to start your stability studies. You need to have a protocol. So making a protocol is very important. If you fail to plan, you plan to fail. It is very, very true for stability studies. So have a clearly defined stability protocol in hand. The protocol should have, first of all, what kind of study you are going to do. Yeah, so this is an interesting part, which I feel everybody misses out when we are reading the API, the Ayurvedic pharmacopoeia. So there are actually real-time studies or long-term, what they've said, accelerated studies, and there's a third kind of study, which is called the cross-sectional study. This is actually a very interesting study design, especially for all the established players in the market who have to now resubmit the stability data for their product. So you've been making it for 10 years now. You have your control samples. So what you can do is you can simply go and pick up your control samples at a 6-month gap. And you want a 5-year shelf life, 3 years. So you pick up for samples from the last 3 years, test them and compile that as a report. And that is good enough. This is a very interesting design, which is permitted under the Ayurvedic pharmacopoeia, and all of us usually miss out on it. So in the protocol, first you decide what kind of study, type and size, number of batches, container closure system, time points, parameters, acceptance criteria, obviously storage conditions and duration. So the API says you need to do 3 primary batches, but it also says you can do pilot batches. You can do 1/10 of the commercial scale. You can take 3 batches of pilot and you can do that. And for the cross-sectional study that I talked about, 2 batches from every year. So if you have a 3 years expiry, you need to pick up 6 batches. So container closure system should be same as what you're going to market. Sampling time points every 6 months in the first year, every 6 months in the second year for the long-term studies. For the accelerated, you need to take 3 points at least. So 0, 3, and 6. That's all you need to do. Test parameters. Okay, so not just assay, but any attribute which can change. So this is a very important point. I've seen stability protocols where people are doing heavy metals at every time point, doing pesticides at every time point. So these kind of things which are not changing, you don't really need to do. So physical, chemical, biological, microbiological parameters, and also preservative content. This is very important, though not a regulatory requirement. It is mentioned in API that you need to do this, but from a practical in-use stability, it is a very important point to also monitor the preservative content. So the parameters, I think Shastriji covered very well. I'll just skip this. So test methods. So API actually says you need to have stability indicating methods, but I don't know how we can do that. What I would say is that yes, we should make sure that our methods are able to pick up small changes, especially within the significant change definition, which is there in API. Because if it is unable to pick up those changes in the composition, especially for your TLC methods, it would be of no use. And having a validated method is very important because you don't want your method variation to be larger than the stability variation. You will never be able to pick up ये testing गलत हुई है या फिर हमारे method में problem है या sorry product में problem है. So how do we do it? So we start a discussion. We as in our lab, so we'll have a discussion with the client. We decide what market, what is the reason for doing the stability, understand the product. Then we look at the relevant regulatory requirements, prepare a draft protocol based on this. The draft is approved, and then after approval of the protocol, we start the study. I recommend you also follow this, step-by-step guide, and only start studies after having the protocol in place. Sample management is very important. Very often you'll see in your team somebody will forget there was a pullout. So it's a good idea to automate it. You can use something as simple as mobile reminders, Outlook, Google Calendar. We use a specialized software for this, and samples should be analyzed as soon as possible. And also it's very a good idea to have control samples. So you put a sample which is 10 degrees below your testing storage, or you can even keep it in the fridge, because when you're in doubt, it's good to analyze a control sample at the same time. So you can really see if there is a variation. Data evaluation. So if you have a 6-month study passing, there is no significant change, you can give 24 months. And obviously you have to follow up with the real time. So what is a significant change? Again, API has done a wonderful job defining the significant change for us. So for AYUSH products, they've said that assay variation of 20%, appearance of new spots in TLC, disappearance of a spot, physiochemical parameters varying by more than 25%, failure to meet acceptance criteria for individual monographs, failure to meet acceptance criteria for appearance and organoleptic parameters. So any of these changes, if they happen, there is a significant change and your study is failing. So you see there is a very generous variation allowed looking at the complexity of our products. So 20%, and these things are quite achievable. And again, your methods need to be up to the mark. So a significant change is again very well defined for passing and failing of the study. So I'll skip the stats part, stat variations. I'll just move on. So again, during the studies, you'll often see you will not always get a straight line. So ideal is that here, if you look at identical slopes and intercepts, this is the ideal study. But usually there will be some divergence. So you need to be able to statistically prove that the batches are similar and understand if it is a batch factor, storage factor, or a combination of those. Sometimes regulatory bodies in CIS and more stricter regulators do ask for these things when they look at the data. So that's it from my end. I tried to very quickly cover the entire thing on stability, and I think thank you so much. You are welcome to email me if you have any more questions.
Wonderful, wonderful, Dr. Saurabh. I think very excellently put. I really loved your first slide itself, which was practically the first slide of a set of gold bars, which everybody wants to keep, and not the rusted pipes, and a very nice example to give. So great going. Thank you so much. I'm sure it's making changes in our thinking of more than about 160 people still logged in and listening. That's great. Time for me to invite Mr. JP Sinha, Vice President of the Analytical Division at Multani Pharmaceuticals. We know one of the best products have been coming from Multani Pharmaceuticals. I've grown up on Kuka cough syrup for many years, starting from my childhood. So I'm sure you're doing some fantastic, I think, quality control back at Multani. So over to Mr. JP Sinha.
Thank you, Mr. Vivek. Thanks for calling me. And good evening to all the dignitaries, Dr. Shastri, Dr. Pankaj, Mr. Sanjay Agrawal, Sri Pradeep Multani ji, Varchastri ji, and Sri Jitendra Sodhi ji. Desktop. Before I start the presentation, I would like to give some background about the testing. In recent years, more and more people throughout the world are turning to use medicinal plant products in healthcare system. Worldwide need of alternative medicines has resulted in growth of natural product market and interest in the system of medicines. On the other side, testing and quality control activities increasingly becoming more and more important in enhancing the trust in stakeholders. The variability of the active compound concentrations, microbial infestation, and residual contamination of pesticides, heavy metals, antibiotics, aflatoxins, etc. in herbs or herbal preparations, which are made of natural sources, are due to genetic, cultural, and environmental factors, has made the use of herbal medicines more challenging than it would otherwise have been. Herbal preparations are mainly originated from natural sources in which chemical, biological, and genetic diversity have been found due to geographical, environmental, seasonal, and other variations. These variations may also affect the batch-to-batch consistency of the preparations products. Therefore, testing and quality control has to play and has been playing an important role in streamlining the standardization and eliminating above-mentioned variations as well as batch-to-batch deviations found in herbal Ayurvedic products. Testing and quality control thus becomes the most important factor in standardization of these preparations. Proper integration of modern analytical techniques and testing protocol development is an essential part in current scenario. Testing protocols include different types of test parameters which belong to physical, chemical, instrumental, and microbiological categories. Testing parameters are performed and methods are developed with the help of modern analytical and high-end instruments. Testing protocols include different types of test parameters which belong to physical, chemical, instrumental, and microbiological categories. Testing parameters are performed and methods are developed with the help of modern analytical and high-end instruments. The testing protocols for AYUSH products. Next. Is it visible on the side? Protocol for testing of Ayurveda, Siddha, and Unani products contain various parameters of testing, the different categories of single and compound formulations. Testing protocols for AYUSH products were developed by Government of India, Department of AYUSH, Ministry of Health and Family Welfare, Pharmacopoeia Laboratory for Indian Medicines, Ghaziabad. The AYUSH products have been divided in major categories of single drugs and compound formulations. Formulations have been divided in physical categories of solid, liquid, and semi-solid type. Testing parameters categorized in 5 major types: organoleptic, biological, chemical, physical, and botanical. Organoleptic, as it is quite evident, there can be different colors of the products: greenish, grayish, yellowish. Similarly, other can be different taste, texture, and fracture. Fracture and the stem and root can have the broken parts, that is called fracture, can have even or uneven. Botanical parameters have got 2 categories: macroscopical and microscopical. Macroscopical, we check color, odor, size, shape, surface characteristics. In microscopical, we check transverse sections, powder microscopy, stomatal index, Palisade ratio, when index number, Wien termination number, lycopodium spot method for start gains. Microscopical parameters, it is quite visible here, transverse section of tulsi leaf by, it is just example we have given to make it understood by one and all. Physical parameters, again, moisture content, foreign matter, flow properties, and particle size, viscosity, diffractive index, extractive values, and ash values. I mean, the method we should mention here: moisture content we do by oven drying method, ash values by furnace ashing method, extractive values by gravimetric process, refractive index by refractometer, optical rotation by polarimeter, viscosity by rotational viscometer, powder flow by Carr's index method, and particle size by sieve method. Chemical parameters, there are different parameters in different categories. First is phytochemical, in which chemical, titration, gravimetric, spectroscopy methods are used for phytochemical analysis. Chromatographic, we use TLC, HPTLC, GC, HPLC, and mass spectroscopy methods for biomarker identifications. Heavy metals and mineral content we do by AAS, that is atomic exhaustion spectroscopy, or ICP OES and ICP MS. Pesticide residue we do by GCMSMS or LCMSMS methods are used for quantification of OC, OP, and pyrethroids pesticides. Aflatoxins we do by SPTLC, HPLC, FLD, and LCMSMS method for residual quantification of B1, B2, G1, G2 aflatoxins. Radioactive scintillation detectors and alpha and beta counters are used for detection of cesium, strontium, beta, and gamma emitters. Identification of phytocompounds by chemical method like alkaloids, flavonoids, tannins, glycosides. Quantification of quality parameters like acid value, saponification value, reducing and total sugar content, peroxide value, etc. Quantification of polyphenol, tannins, sugar, and UV visible scanning by UV visible spectrophotometer. Fingerprint scanning and quantitative analysis by biomarkers by HPLC. Quantitative analysis of biomarkers by using SPLC technique. Analysis of heavy metals, mineral compounds by using AAS technique. Analysis of trace level of heavy metals, minerals, and other metal components by using ICP OES and ICP-MS technique. Analysis of pesticides, aflatoxins, and other natural contaminants by using LCMSMS technique. Analysis of radioactive contamination by using scintillation detectors. Analysis of alcohol content, pesticides, and residual solvents by using GCMSMS and GCHS FID technique. Biological parameters, we can categorize different categories. Microbial MLT test, total aerobic bacterial count, and total yeast and mold count. Then pathogen detection, E. coli, S. aureus, Pseudomonas, and Salmonella. Pharmacological screening, anti-inflammatory activity, anti-diabetic activity, etc. Toxicological screening we can do by acute toxicity, lethal dose calculation, or severe toxicity by histopathology of body organs. Enumeration of total bacterial and total fungal count by using microbial limit test techniques, detection of pathogens in AYUSH drugs by using selective media growth and biochemical test methods, in vivo and in vitro methods for evaluation of pharmacological effects, acute and chronic toxicity evaluation by using in vivo animal models. Now this is a table, you can clearly see there are a list of parameters which are used for different kind of AYUSH products, be it Arca, Asav, Arista, Netrabindu, Syrup, or Avaleha. What parameters are applicable in which kind of products can be determined by seeing this table easily. So I'm skipping this slide. Similar is the case in next stage. Different products are different. I mean, ASU products are shown here with different parameters. Which parameters are applicable in which kind of products? I'm skipping the slide. Same is the case here. I'm skipping the slide. Now we come to the stability study of AYUSH products. The purpose of a stability study is to establish a shelf life and level storage instructions applicable for all future batches of the drug product manufactured and packed under similar conditions. The shelf life thus determined can be used to decide expiry date in case a manufacturer wishes to declare a shelf life longer than one specified by the notification substituted by GSR 789E dated 12/8/2016, which is from 12/8/2016. What is stability? The ability of pharmaceutical or AYUSH products to retain its physical, chemical, biological, and microbiological properties within specified limits throughout its shelf life. Or, in other words, the ability of pharmaceutical AYUSH products to retain its quality, safety, and efficacy throughout its shelf life. Types of stability studies. Although it has been, some details have been given by Dr. Saurabh Arora and Sri Shastriji, still we have to mention some points here. Accelerated studies designed to increase the rate of chemical degradation and physical change of a drug by using exaggerated storage conditions. Real-time stability studies experiments on the physical, chemical, biological, and microbiological characteristics of a drug during and beyond the expected shelf life and storage periods of samples under the storage conditions expected in the intended market. AYUSH Guidelines for Stability Testing, Ayurvedic Pharmacopoeia of India, Part 1, Volume 9, 2016. The objective of this guideline is to specify the method of arriving at shelf-life by stability testing. The shelf-life determined by the process mentioned in this guideline can be used to decide the expiry date in case the manufacturer wishes to assign a shelf-life longer than one specified by the notification GSR 789E dated 12/8/2016. The guideline can be used for all patented and proprietary Ayurvedic medicines, both new and existing products. Now, the general information on stability: selection of batches, container and closure system, specification, testing frequency, storage condition, and evaluation. When is the stability study required? Stability studies shall be done on 3 consecutive batches of each new drug product and or if any changes in vendor of active ingredient, formulation of the product, manufacturing procedure of the product, primary packaging material of the product. In addition to this, first batch of every product should be yearly taken for ongoing stability studies program. Factors affecting stability. Number 1, environmental factors, ambient temperature, humidity, and light. Second one, product-related factors, physical and chemical properties of API and excipients, dosage form and its composition, the manufacturing process, the nature of container closing system, the properties of primary packing material. Storage conditions. There are 4 climatic zones worldwide, and we belong to the zone 4, hot and humid. And that is why we will take this condition only at this present. Recommended storage conditions in this zone are: number 1 is accelerated, the temperature 40 degree plus minus 2, and relative humidity 75% plus minus 5%. Minimum time 6 months. The second one is real time. The storage condition temperature 30 degree plus minus 2 degree and relative humidity 65% plus minus 5. Minimum time 12 months. Selection of product batches. Stability studies are conducted on at least 3 primary batches. For new products, pilot batches are used, which are at least 1/10 of the commercial batch size. Possibly, batches of drug products should be manufactured by using different batches of drug substance. Sample size or quality of quantity to be selected in a way which will be sufficient to test in duplicate for each time withdrawal. Requirements of stability studies. Stability studies protocol, stability studies planner, stability study chambers, calibration and mapping records, stability study chambers, temperature and humidity monitoring records, daily monitoring, temperature and relative humidity monitoring twice a day or online temperature and RH system, temperature, thermal mapping and RH calibration once in a year. Testing frequency. For accelerated studies, it should be initial stage after 3 months and 6 months. For real-time studies, it's at initial time, 6 months, 12 months, and then once in a year. Tests required for stability studies. Tests to be carried out for accelerated stability studies in solid dosage forms, tablets, capsules, and powder. These are the parameters: moisture, LOD, pH value, weight, variations, extractive values, ash values, particle size, hardness, friability, wherever applicable, TLC, assay, microbiological limit test, and detection of specified pathogens. Tests to be carried out for accelerated studies in liquid and semi-solids. Description, total solid content, pH value, sugar content, preservative content, if any, TLC, assay, microbiological limit test, and detection of specified pathogens. Tests to be carried out for accelerated stability studies in grit and tela samples. Description, iodine value, saponification value, effective index, peroxide value, rancidity, acid value, TLC, assay, microbiological limit test, and detection of specified pathogens. Tests required for stability studies. Tests to be carried out for real-time stability studies in solid dosage form, tablets, capsules, and powder. The same parameters are here, only the frequency has been changed. So I'm skipping the slide. Again, it's the real-time parameters are same, only the frequency has to change. So I'm skipping the slide. Skipping the slide. Now, stability study, results and interpretation. Significance. An Ayurvedic drug can be considered to be stable if no significant change occurs at any time during testing at accelerated storage conditions or at real-time storage conditions. Stability study results and interpretation. What is significant change? Now it has been defined as per API. We can check it. A plus minus 20% change from the initial assay value if the drug is analyzed for its marker compound. Second, a plus minus 15-degree change from the initial assay value if the drug is analyzed for its active compound. Number 3, appearance of new spots in identification by TLC when compared with the sample stored in less than 10 degrees. Variation in the physiochemical parameters, moisture, as particle size beyond 25% of the initial value. Failure to meet the acceptance criteria as per individual monographs or specification. Failure to meet acceptance criteria for appearance, physical attributes, and functionality tests, e.g. color, phase separation, caking, hardness. Importance of a stability study. Introduction of this has been found very important in the market today. Introduction of newer drugs to the healthcare market. Stability testing ensures compliance with international regulations that form part of the registration process for a new drug, substance, or drug product, determining the safety and integrity of the drug product, and also its shelf life and storage conditions. Ensure validation of product manufacturing process, ingredients used, packaging materials, and desired shelf life. Increase the product acceptance among diverse customers. These are the references which we have taken from sources. So I'm skipping this slide. I'm just giving a brief about Multani Pharmaceuticals Limited Laboratory, which we are running here. We are approved, Multani Pharmaceuticals Limited and Analytical Division is approved for the testing of Ayurvedic and Unani drugs by the Directorate of Ayurvedic and Unani Services, Dehradun. It is approved for the testing of drugs and pharmaceuticals by Drug Controller and Licensing Authority, Directorate of Health Services, Dehradun. NABL accredited as per ISO IAC 17025-2017 for chemical and biological fields. Scope covers the testing of drugs and pharmaceuticals as per IP, BP, USP, cosmetics and essential oils, homeopathic drugs, Ayurvedic, Unani products, and food and agricultural products. The laboratory is designed according to the current EHS norms, environment, health, and safety. Chemical, instrumentation, and microbiological sections are well separated to avoid cross contaminations. Microbiology section well designed with 3 AHUs and other essential equipments. Team of well qualified professionals, trained internal auditors as per ISO IEC 17025-2017, equipped with advanced techniques, instruments according to current regulatory and statutory requirements. Well-defined system in place to handle conflict of interest. GLP compliant, certified by the Office of Drug Controller Uttarakhand. Use of proper CRMs, related substances, impurities, working standards, intermediates to ensure quality results. Multani Pharmaceuticals Limited Analytical Division has been supporting analytical research to government and private sectors in most viable way. Support to academia, research projects, training programs for students and research scholars for proper quality control in ASU, pharma, cosmetics, and food industries. Dedicated support to Ayurvedic and Unani and pharmaceutical drugs, promotion programs across the borders by developing the analytical methods and their validation, stability studies, and by controlling the contaminants. Providing training to needy personnel on a very nominal fee with an aim to enhance their skills. Our equipments at a glance, we are having at present atomic absorption spectrophotometer AAS with FLEAM, GTA, and VGA. High performance liquid chromatograph HPLC. We are having 10 numbers with RI, FLD, UV visible, and PGA detectors. FTIR we are having with ATR. UV visible spectrophotometer and gas chromatography. We are having 4 numbers with HS and auto liquid detectors. HPTLC applicator with photo documentation, double door autoclave, vertical autoclave, BOD incubators, closed system for stability testing, Carl Fisher potentiometer, auto titrator, resolution tester, 4 numbers, manual and auto sample. Laminar airflow, 2 numbers, biosafety cabinet, liquid optical particle counter, stability chambers, accelerated and real time, 3 numbers we are having at present. Soon we are upgrading our infrastructure and facility with high-end and modern analytical instruments like LC-MS-MS, GC-MS-MS, and ICP-MS, RT-PCR, and many more to support the ASU and food products. We are also coming up with a new walk-in chamber stability room dedicated for the stability studies of ASU projects. With this, I will in the program right now, and I will again thank to all PhD CCI members, the office bearers, to giving me this opportunity to put my views here. Thanks to all.
Thank you very much, Mr. Sinha. Thank you for a wonderful presentation and giving us the details also of the analytical division, so that a lot of members and manufacturers out here can also take the benefit of your laboratories. Thank you so much. Truly revealing, truly revealing. Time for me to call our last speaker of the day. It's none other but Dr. Arvind Tripathi. He is the manager for quality control at Hamdard Laboratories, yet another very reputed organization, brand. And as I think Dr. Saurabh Arora had said, brand versus profitability, you know, I think that balancing, if we have to talk about, we can always talk about a company like Hamdard Laboratories. I still, you know, enjoy your Ruh Afza, and I just had it minutes before our meeting started to refresh me after I came from the vaccination center. So over to you, Mr. Tripathi, for a presentation or a talk as good as your Ruh Afza.
Thank you. Can I get the access to share my content?
Sure. So can I request the admin or the host, Paramjeet, to kindly give him sharing rights for his presentation?
It's already given. He can share the PPT.
Thank you, Paramjeet. Thank you so much. I hope, Mr. Tripathi, you are able to share. Yes, Dr. Tripathi, we can see your screen.
Okay, now I start my presentation.
Please go ahead, please.
Okay, just first of all, I will just give the thanks to all of our members, Honorable Dr. Jalen Sastry, CEO MMPB, Dr. Pankaj Goyalji, Director MMPBL, and Dr. Pradeep Multani, Senior Vice President, PhD Chambers, and all respected panelists, and all leader team members of PhD Chambers and participants. Good evening, and I'm Dr. Tripathi representing the Hamdard Laboratory India Medicine Divisions. We are really thankful to PhD Chambers for taking all initiative for uplifting AYUSH system of medicines under the able leadership of guidance, Sri Pradeep Multani ji, and organizing such a webinar, which gives the insight about strength and gap in the system. We are thankful for brief elaborations and importance of self-life by Dr. Jalen Sastry. In fact, we are continuous learning. It's always a dream for any AYUSH drug manufacturers to get the whole COPP certifications for their products and thereby achieving high quality of standards, enabling them to export their products under the regulations. Self-life is a part of requirement of such certifications. So here
we're not able to see your PPT.
Yeah.
Just could see the screen. Yeah.
Okay. Just I'm just take a little bit time because already Dr. Saurabh and JP Sinha and Dr. Jalen Sastry has already explained to all of detail about that modern self-life studies. So stability study in the modern science and adapted in Unani systems is just I represent here. And here just I focus that nobody can cover this self-life. As per Drug and Cosmetic Act in the Unani systems, according to Drug and Cosmetic Acts, here slide is represented for the yellow colors, which is according to Drug and Cosmetic, the self-life is already increased in these products segment category, especially the halwa category, jawarish category, fusta and log. And other hands, marham and jamar, two categories in Unani systems where self-life is already decreased by the Drug and Cosmetics Act. And mufarre is already increased and tila is already increased. So here the stability testing methods, two types of generally methods already explained by Dr. Sastry and Saurabh and all things. Just I give the in very short manner. Stability testing methods have generally taken the accelerated and real times. Sometimes it will perform in intermediate testing for 25 degree, which is not considered in our AYUSH system. And stress conditions for force degradation system. It's generally in stress conditions we have covered under the accelerated conditions. And force degradation studies, you can just studies that the possible degradation product in your formulations. And stability depending on the various factors like stability information is not a correct self-life. It is the expiry date, mandatory requirement for all license, Ayurvedic Unani system of products. Stability depending on the various factors like the nature of products, the ingredient of the products, and packaging materials also. So stability studies are carried out to demonstrate the medicines will remain suitable for consumption during the shelf-life period, which is storage under the conditions mentioned on the packaging. On product label, there is no mentions about that any specific storage condition. Then it will be assumed that the storage at the room temperatures. Generally, we have taken for 30 degrees Celsius. And factor influencing during the stability studies, there are generally taken the four factors is temperature, humidity, and storage place. Above factors to establish the retest period of the drugs or the shelf life of drugs products. And storage condition is already defined as per ICH guidelines, and we have already divided the 4 climatic conditions for the performing of the stability studies. So, if on the basis of climatic conditions, we have recommended the 2 major storage conditions: stability for accelerated studies, that is performed at the 40 degree plus minus 2 degrees Celsius and 75% RH plus minus 5% RH. And it's performed 3 and 6 months, basically the accelerated conditions. And the long-term conditions will take place for 12 months. And different storage conditions recommended by different bodies like ICH and WHO, where we have just only already presented by the Dr. Saurabh. And according to the schedule for samplings, we have just planning your protocols for the 6 months, 9 months, 12 months, 18 months, and 36 months, according to depending on the shelf life of your finished products. You can plan the stability studies. For evaluation of stability, there are the purpose of stability to establish the based on the testing minimum of the at least 3 batches and retest period application for all batches during drugs substances of the shelf life, level, storage instructions applicable for the all future batches of the drugs products manufactured and packed under the similar circumstances, and Ayurvedic Unani drugs can be considered to be stable if significant changes, and significant changes already explained by Dr. Saurabh very nicely, but on here just I give the 1 points for the significant changes. If the active assay value is at the initial stage, and if you consider the marker that is considered under the 20% of the changes, and where if you have to check the analyze for the active compounds, especially, then it will be considered under the 15% for the significant changes. So acceptance for the new spot in identification by TLC when compared with the samples stored in the less than 10 degrees Celsius are completely disappear or not existed on spot. when you perform the TLC. So that times you can say that this is a significant change and consider another significant changes. So failure to meet the exception criteria as per the individual monographs or the specification, and failure to meet the exception criteria by the appearance, physical attributes, and functionality testing like color, face separation, cracking, and hardness. All these types of the parameters can be evaluated in our different categories of products.
Dr. Tripathi, if you are not showing the presentation, you can share the screen so that we can see you in a bigger icon on our screens.
Okay. So let's have just last 2 slides focus for that one. Basically, the stability studies in modern science and what is their challenges. We have focused there are the 4 and 5 points: lack of instrument analysis and development for uniform punctual methods not adopted by the industrial level, and as herbal formulations are complex in nature and multiple ingredients mix poses the problems in chromatographic separations. Authentic source of raw materials availability across all season, that is also the major role to not important factors, and non-availability of active markers standards, appropriate method for evaluating the traditional medicines, and clinical trials facility not easily available along with the stability sample analysis at industrial level. SPTLC fingerprinting is the more feasible to evaluate the marker compounds in the complex matrix formulation of drugs, but HPTLC have preferably techniques for evaluating the single active principles and their acceptability in the science community. And finally, they conclude that self-life, as described in Drug and Cosmetic Act, need to be validated with appropriate analytical techniques through stability studies, and the self-life is greatly depending on the quality of packaging materials and also storage conditions. Proper rationale need to be established. Self-life of products like Fusta need to be further established, as these are very stable, and self-life is near about 10 years. Since herb source are not depleting, hence there is a need to have regulatory provision, that is if a product is complies to specifications beyond the shelf life, it can be marketed with extended shelf life proof. So finally, I conclude my views. Thanks for panelists and all the leaders and team members for PhD members. Thanks a lot.
Thank you so much, Dr. Tripathi.
Thank you.
I think very well explained, minus the slides. I think you did a very great thing by, not covering all the points which were already covered, and therefore, coming right to the point and making it very, very informative and interesting. So thank you very much for your talk. I'd like to invite any questions, because I just see appreciation in the chat box for all the speakers and not really a question. So we can give 1.5 minutes to the audience to put in their questions if they have any. And if they don't, because they have got all the information, it'd be good to conclude with a formal vote of thanks. So we still have close to 100 attendees there, and in case you have any questions, quickly type them into the chat box, unless you've got all the information you needed. Thank you, Milanji, for that compliment, which you just put into the chat box. And similarly, a lot of other attendees have also put in their compliments. And I'm sure, all of us are going back home today a lot more informed and with lot more knowledge about the protocols and the stability studies. Okay, I just seem to be getting only compliments and no questions. Okay, there is one question. So thank you, Mr. Ravindra, for all those compliments as well. Question to Dr. Piyush. Many customers are accepting NABL reports in USA. Is ILAC not applicable in USA? Over to you, Dr. Piyush.
For Dr. Pankaj.
Yeah, I'm so sorry. Dr. Pankaj, I just read verbatim what's written there. So my mistake, Dr. Pankaj, over to you.
Sir, actually, question is not visible to me. Can I have the question again?
Yeah, I'll just read it out for you again. It says, many customers are accepting NABL reports in USA. Is ILAC not applicable in USA?
Well, sir, ILAC it is the International Laboratory Accreditation Corporation. It covers whole globe, whole universe under its umbrella. So ILAC is the coordinating body for throughout the world, all the economies. Around 87 economies are there right now. In Asia Pacific region, particularly, there is the APEC body, which is coordinating all the Asian countries. Okay, so ILAC is available in US as well. In US, there is a definitely other accreditation body, like in India, it is a ABL. So in US also, the ILAC is applicable.
Okay, all right. There's a question from Milanji here. I wonder if somebody can answer that. Maybe the industry people may have a good idea on this, or even Dr. Gaurav. Any subsidy shall be arranged for micro industries as the cost of compliance to stability studies is higher for micro industries, which form the majority of the industry. So I think the essence of his question is that I think we can hear some background noise. Mr. Sodi, I think it's coming from your side. If you can mute yourself. Thank you, Mr. Sodi. So I think the essence of the question of Mr. Milan Mehta is like any subsidy which is available for these stability studies, especially for MSMEs, as most of us are MSMEs in the AYUSH sector. So would anyone like to attempt this question? Whether it is Mr. Sinha or Dr. Tripathi, any idea? I'll request Dr. Tripathi to unshare his screen, please.
Well, I'm JP Sinha. Since it's a question related to subsidy,
yes,
I think the better person to answer this question should be from government body.
Apparently, Dr. Sastry is gone for the meeting with the secretary. So I thought in case industry panelists here have any information they'd like to share, or if NABL has any information on this. Okay. So Milanji, I think we'll come back to you on mail with the answer to your question. And time for me to conclude the program. It's been a wonderful evening. And as I said at the start, it's a fateful evening. Closer to the end of the webinar, we've also closed by vaccinating about 450 people, making them safer. And this has been a very interesting webinar on testing protocols and stability studies of AYUSH products. And we'd like to thank in absentia Dr. JLN Sastry, the CEO of NMPB from the Ministry of AYUSH, Government of India. Dr. Pankaj Goyal, not in absentia. Thank you, Dr. Goyal, for staying back for the full length of the program, Deputy Director of NABL. I'd like to also thank our vibrant speaker, Dr. Saurabh Arora, the Executive Director for our Analytical Division, Arbro Pharmaceutical Private Limited, Mr. JP Sinha, Vice President of the Analytical Division from Multani Pharmaceuticals Limited, and of course, Dr. Arvind Tripathi, Manager for Quality Control at the Hamdard Laboratories. It's been tremendous learning, I think, for me personally and for a lot of us here. who've been attending the webinar. And I'm sure it's going to help the industry improve the standards of the product through these testing protocols and stability studies information which we got today. I'd like to also thank our Senior Vice President who's had to leave for a meeting, and he texted me that for being here and guiding us and motivating us. I'd like to thank our chair, Shri Arjun Varchasvi ji, who also had to leave because we extended beyond our time. And I'd like to thank our co-chair, Mr. Jitendra Sodhi, for staying back for the length of the program. And it's been excellent having all of you here today, and look forward to having any and all of you next time again in our webinar, which we perpetually keep doing almost every fortnight. So thank you very much. Have a nice evening and stay safe with your family, friends, and relatives. Thank you so much. Bye-bye.
Thank you, sir. Thank you very much, Vikti. Thank you, everyone.
Thank you very much, sir.
Thank you, Saurabh-ji.
Thank you very much, sir.
Thank you so much, sir. Thank you, everybody.